Hi, hello, everybody. It's nice to be back again, and we are in the afternoon session of the conference, and I'm very honored to introduce you my friend and colleague, Dr. Suan Chu from University of Texas, Rio Grande Valley. This is the second keynote of our conference, and I'm very happy to be here to chair this session and to listen this keynote with you. So Suen, please, you can take the speech.
Thank you so much, Serena. Let me share here. Okay, can you guys see my screen all right? Can you guys see it okay? It's good? Sorry, Serena? Okay.
Yes.
Okay, I wanted to make sure. Thank you so much, Serena, for inviting me. I'm very thankful to Serena, to Pascal, and also to Ugo for just hosting this great conference when it is such a difficult time and still gathering us to share our work. Thank you so much. So, like Serena said, I'm from University of Texas, Rio Grande Valley. And today I'm going to share with you our work on PLGA scaffolds fabricated with an easy and economical moldless techniques for different application. So just to share, Serena and I met quite some time ago when she visited Texas to be a visiting researcher with us in the MECOS lab. So I did my PhD in the MECOS lab in bioengineering. That's where I met her, and then after that, I went to Baylor College of Medicine, just across the street in the medical center for my postdoctoral work. And then I moved all the way down to the lowest tip of Texas, where it's UTRGV has one of its main campuses. It's called Brownsville, Texas, and there I was. I am in the I am right at the border of Mexico, and Serena has come to visit me already twice, and I was hoping this year I could be there with you guys to visit Pisa finally, but this happened, so next time. So you guys know about PLGA. Polylactic glycolic acid, it's very common. It's FDA approved for many applications. It's often used because it's biodegradable, biocompatible, and the degradation can be tailored by changing, the lactic and co-glycolic acid ratio, or the tips, the functional groups of the tips, or the molecular weight of the tips. And scaffolds are usually used, for many applications, such as tissue engineering or cancer therapy, for structures that can provide temporary place for the bioactive factors of the cells that we are trying to deliver, and it will also help with the localized delivery of these components. So when we started the laboratory in Texas, in Brownsville, we didn't have much material. So we wanted to, not much funds, not much material. So we wanted to think of a technique for us to make scaffolds that I could get my students, which is a predominantly undergraduate institution. How can we get us starting with working with our scaffolds right away? So we thought about, we looked around in literature. We saw a paper by Krapp et al. From JBMR Part A in 2011, where they use a injectable scaffold method that has a moldless, prefabricated, moldless scaffold method where they don't need mold. It's very easy and economical. So we started exploring that, and we tried to improve what we learned from there. So we started by making different architectures, different surface that leads to different surface area to volume ratio, and that also leads to different degradation rates and release kinetics. So just a little bit how this moldless technique work. We have to start with a solvent called tetraglycol. It is biocompatible, and we dissolve our polymer our drug into there. After it's dissolved overnight, we use a syringe or a different single pipette tips to deliver these scaffolds into PBS, a buffer saline buffer solution, where the scaffold will precipitate. So if you see on the right, we were able to make different architectures, and if we collected them at the bottom of the beaker, where when we push it out, it touches or it pushes against the beaker, we are able to form these different globular but different architecture scaffolds, which we were able to compare. So when we started, Krebs et al., when they used this scaffold, they used it for plasmid DNA. They also used the globular one where it has the least surface area to volume ratio. But we wanted to be able to discover or investigate different architecture, and we also wanted to make sure that we were able to sustain the release, right? So we explore how can we increase entrapment efficiency and drug loading with this method that we were trying to use. So, after we inject the scaffold into the PBS solution, it will precipitate. So the tetraglycol will come out of the solution into the PBS, and then we moved it into different solution to further precipitate it. So, we tried different things. We tried PBS, we tried methanol, we tried ethanol. And if you see in the graph, the one on the most right, we saw that when we use ethanol, we were able to increase the amount loaded and also sustain the release. And we believe this is so because the PLGA precipitates very well in alcohol. So, by going from a PBS solution to the ethanol, we could further precipitate the polymer out of solution and trapping the drug inside. Okay, so we made the different architecture, the globular, the thick strand, and the thin strand, right? Globular was with the syringe, the thick strand was with the 1000 ml single pipette, and then the thin strand with the 200 micropipette. So we can see here that the surface area and volume ratio could be changed based on that. Something that is thin results in higher SVR, surface to volume ratio, and which we believe that can lead to faster degradation, right, because of the more surface area, and also a faster release of the drug. So then the other thing we wanted to make sure we do as we characterize was to make sure that we check the percent drug loaded and entrapment efficiency. And again, this is also based on the SVR, right? And when you have a scaffold that is like the globular, where you have low SVR, we were able to have a higher entrapment efficiency and higher percent drug loading because there were less surface for the drug to get lost during the fabrication process, right? So the next thing was scaffold degradation. We wanted to see how these different architectures could lead to different degradation rate. We were expecting that the tin would degrade faster. We were thinking, okay, this SVR was the highest. This means that more water molecules can get to the ester bonds on the PLGA and degrade it. But the thing is, we saw the opposite, right? We saw that the globular was the one that actually was losing more of its weight over time, and we looked into it more, and we saw that this occurs because when you have the globular scaffold, you have your acidic byproducts that are stuck in the scaffold, right? That lactic acid, the glycolic acid. So that instead will auto-catalyze the degradation of the other ester subunit. So that's what we were seeing. The entrapment of those acidic byproduct led to that. So that's why this opposite effect that we're seeing. And I expect that, for you guys who are familiar with PLGA, we can change the L and the G, right? The 50 and the 50 and the 75 and 25. We can change that ratio to be able to tailor the degradation. So the PLGA 50-50 has more lactic acid and it's able to degrade faster than the 75-25. So the next thing we looked at was drug release. we wanted to see how changing this SVR would change the degradation rate, right? Not the degradation rate, the release kinetics of the drug. So here, we saw that the thin one, was able to the globular one again was, interestingly for the 75-25, it was releasing more drug. And at the 50-50, we were able to see a biphasic release profile, right? It was a burst release in the beginning, a small burst release in the beginning, which is expected for scaffolds, right? But and in the middle, we see another burst release. So it was based on how that scaffold was degrading, and the morphology changes that we see that second biphasic, that second burst release. Again, we saw that the surface volume ratio was not the only thing that was making or controlling the drug release. It was also tied to the degradation of the scaffold, right? So it was not a clear-cut understanding of how it happens, but it's a mixture of both of these, right? The degradation and this surface volume ratio. So, in conclusion, for this part of our study, we saw that scaffolds with different surface volume to area to volume ratio we were able to fabricate it, and it can result in different degradation rate. We saw that the accumulation of the acidic byproduct led to lower SVRs, having the autocatalytic degradation. And as expected, we saw that scaffolds fabricated with 50-50 degraded faster, and this leads to more drug release over time compared to 75-25. And then one of the ones that we saw interesting was the PLGA 50-50 globular scaffold. We resulted in a biphasic release profile. So a burst release in the beginning and then midway, which can be useful for certain applications. So where did we take this scaffold next? So this is some of the current work, that my lab is doing to see how we can use this scaffold. So the first one, we are looking at investigating the utilization of iron oxide nanoparticles to control the release of a specific drug from these scaffolds for the treatment of glioblastoma. And then a second project that my students are working on is the use of these scaffolds for a dual delivery system. So we are looking at a system to deliver metformin and Y15 for the treatment of platinum-resistant ovarian cancer. So now I'm just going to go over what we're currently doing with these projects right now. So first, the glioblastoma model that we're using. It's a very aggressive malignant brain tumor, and it's linked to low relative survival rates, with only 6.8 of the patients surviving 5 years post-diagnosis. So there is definitely a need to find a treatment method for glioblastoma, and we're working with a collaborator in the physics and astronomy department, where we are utilizing iron oxide nanoparticles. These particles are able to generate heat when a magnetic field is applied to it. It's so far shown to be non-toxic, and we are also using these particles that are dextran coated. So the dextran allows the prevention of the sticking of each particle together as they grow closer to one another, and they stick. That can cause more toxic effects. So we make sure that we're using the dextran coated one. So how it works is we have the scaffolds, and we use the scaffolds, but we use the one that was the globular scaffolds because we found that one we were able to more easily take in the nanoparticle and the shape of it and explore it. So that scaffold right now, we're making it with different amounts of nanoparticle. We have 5, 10, 15, and 20%. And when we give it an alternative current, so AC magnetic field is provided to the scaffold using a magnetic hyperthermia machine, we see that because of Brownian relaxation as well as neo relaxation, the particles can provide heat and can heat up the scaffold. So the drug that we're using for this glioblastoma model right now is minocycline. So minocycline is well known to be an antibiotic drug for bacterial infection, but right now it's also being explored as an anti-angiogenic effects. Its angiogenic effects on tumors, and glioblastoma is one of them. So how it works is it has the ability to inhibit matrix metalloproteinases, and these MMPs are important for angiogenesis. So by inhibiting these MMPs, we are able to inhibit angiogenesis. So in the long term, we're looking at the minocycline combination with a chemotherapy drug, temozolomide. But this work we're exploring right now is just looking at the minocycline part. So, for this work, our investigation was started with looking at the effects of the different concentration of the iron oxide nanoparticles. And there we wanted to relate it to the effects on drug release rates and then study the effects on cell viability. So this is just some macroscopic images of the scaffold. The iron oxide nanoparticles are very dark. It's brownish, blackish looking. And as you add more, as expected, we get a darker scaffold. Right now, we're trying to get SEM images. We're having trouble because sometimes the scaffold is still wet or it still has tetraglycol remaining in it. So we have challenges with that. So if you guys have any ideas, do tell me how to look at these scaffolds in SEM. So, we wanted to characterize them with drug loading and entrapment efficiency. So we were able to make sure that the scaffolds were generated evenly and repeatably, where their entrapment efficiency remain consistent for all group. But as expected, if we put in more nanoparticles into the scaffolds, the drug loading percent goes down because the iron oxide nanoparticle concentration go up. We had to adjust for the minocycline that it's actually injected with the scaffold. So the first thing we looked at is how changing the amount of iron oxide nanoparticle changes temperature over time, right? Remember when we have the iron oxide nanoparticle and we provide that magnetic field, we can heat up the scaffold, right, or the surrounding area. So we saw that when we increase the amount of iron oxide nanoparticle, we were able to generate more heat, and we were able to increase the temperature, which is we were targeting a change from 37 degrees Celsius, which is physiological temperature, to 42 degrees Celsius. So a change of 5 degrees Celsius. We were able to do that at a faster rate when we are using more iron oxide nanoparticle as expected. We are targeting 42 degrees Celsius because that is what is usually used for hypothermia in cancer, where they use heat to kill cells. So we were trying to make sure we kind of remain in that temperature range, and to show that it's not toxic to the surrounding cells that are not cancerous. So the next thing we did was we wanted to see how the drug release is affected or changes when we go from a 37 physiological temperature to an elevated temperature of 42 Celsius. And as you can see here, the blue bar, you can see that there was a increase in amount of drug that gets released compared to the 37 physiological temperature. So, as expected, we are able to control the amount of drug getting released when we provide a heat to the scaffold, and this is due to the PLGA polymer changing from a more solid state to more of a liquid or more of a mobile state. The chains are moving around more, creating this void in the polymer scaffold, and the drugs are able to release. So it's all about the transition glass temperature of the polymer, and based on that, the release will be different. And so the next thing we wanted to do is kind of look at how these components or this system react to we will affect the glioblastoma cells. So U87MG cells is a common glioblastoma cells, and it's the one we are also using in our lab. So the first thing, although the literature has shown that this nanoparticle is not cytotoxic, we want to make sure that at the percentage that we were using in the scaffolds, that is not toxic to the cells. We don't want the iron oxide to be the one killing the cells. So we found that they were not toxic compared to the control. And then the next thing we wanted to do is we did um, two cell viability test. The first one is a hyperthermia test. We wanted to check when it's at physiological temperature compared to an elevated temperature. How is it, right? And we saw that the elevated temperature can induce cell death, which is expected because magnetic hyperthermia uses that temperature. And then we wanted to see as does the drug that gets released at 42 degrees Celsius, is it going to kill the cells and also maintain its bio activity, right? We were worried that the 42 degrees Celsius will deactivate or degrade the minocycline. And here we saw that, at the 42 degrees Celsius, we are seeing, again, the drug release able to kill the cells more than the 37. So, our question next is, can we see a synergy in this? Is it the heating of the cells as well as the drug being released from the scaffold? Can we see a synergistic effect? So that's what we're exploring next. So just to conclude, non-toxic nature of the iron oxide nanoparticle makes it a good candidate for us to use. By heating it, we are able to induce hypothermia. We can control the drug release rate, and we can enhance cell drug cytotoxicity for cancer treatment. So the next project, the platinum-resistant ovarian cancer project. So, ovarian cancer is the fifth leading cause of cancer among women in the US. And usually, the treatment method for ovarian cancer is platinum-based drug such as cisplatin. But a lot of the time, the patient will get resistance towards this chemotherapy drug. So, there is much need to try to find a way to treat platinum-resistant ovarian cancer. So, there is a drug called Y15. It's a FAK inhibitor, and FAK is a focal adhesion kinase, which is a tyrosine kinase that can trigger multiple signaling pathways. So it's shown to be, activated in platinum-resistant ovarian cancer. So so this leads to FAK inhibition can be good for platinum-resistant cells. Then we also, wanted to see how FAK inhibition or the Y15 inhibitor, how can we can improve its function when we combine it with metformin. So metformin, a lot of people know it as a anti-diabetic drug, but it's also shown to have some anti-cancer action. So, we wanted to see if, we can combine these two, and this is the work done by my collaborator, Dr. Akin Levy at Nova Eastern, and it's in the process. We're trying to publish this work right now. So, looking at the graph, the first two graph is just IC50 values, a graph where we did a dose response curve to show the IC50 values of the cell of the platinum resistant ovarian cancer cells. These are OVCAR3 cells, and we saw that the IC50 for metformin was around 26 millimolar, I would say pretty high. And then for Y15, it's more sensitive. It's 80 micromolar. I think it's expected. Metformin is an anti-diabetic drug, right? So, you expect it for it to function as a cancer drug. It has to be used at a pretty high concentration. So it can be a challenge sometimes to get to that concentration to have that effect. So over here, in a 2D model, we saw that combining the metformin and Y15, we can get a synergistic effect compared to the solo drugs itself when they're delivered individually. So how it works is, the FAK inhibitor Y15, you would expect it. It's able to, decrease FAK phosphorylation. So, FAK is a kinase. So the way it works is by phosphorylation. But metformin, it hadn't been explored as much before. We saw that it's also able to decrease the phosphorylation of FAK. So, with these two combined, we hope it's can combine and be synergistic and decrease it further. So all these happens with apoptosis. So we saw that DAX and P53, the pro-apoptotic molecules was increased. BCL2, which is the anti-apoptotic molecule, was decreased, and PARP cleavage, which is an indicator of apoptosis, was also decreased for both metformin and Y15, and even and usually the combination we see a stronger effect in apoptosis compared to the solo. So just what my collaborator Arkin Levy, her hard work has shown for this part is Y15 and metformin is able to inhibit FAK phosphorylation and cause that synergistic effect. So we with this work, we wanted to see, let's try combining it with a biomaterial. Let's see if we can improve it, right? So we used that same model we had, the PLGA particles up PLGA scaffolds that we use with the moldless technique. We made different solution for there, just so that you can see how it's done here. Um, and then we inject it into PBS and form those globular scaffolds. So, on the left here, I'm showing you the free drug, the work that was done previously on the 2D culture where, drugs in solution were exposed to the cells, right? There was a nice synergistic effect. But you see on the right, when we release the drugs from these scaffolds that we made, we see an even more improved synergistic effect. I say that because the solo drug itself, we see that there wasn't much effect, right? If we do the statistic, it's comparable to the control. But amazingly, when you combine these two together, we see that synergistic effect. For both, when we use 100% of the release solution or 50%, meaning we dilute the solution with 50% of media, and we still see that synergistic effect. And when I first saw this, I told my students, I'm unsure. I'm excited, but I'm unsure. So they had it. I had them repeat twice, and to confirm that we are still seeing this effect. So just to conclude, metformin in combination with FAK inhibition, shown both in our lab and with Dr. Levy, we could see a synergistic effect, making it a promising avenue for therapeutics for our platinum-resistant ovarian cancer. And the dual delivery of metformin and Y15 using the scaffold we've been discussing here is promising also, and it's a good biomaterial for future for this type of cancer. So just an overall summary, PLGA scaffold fabricated with this easy and economic moldless technique was tested in this project. And it can be promising materials that can be used in combination for iron oxide nanoparticles to be able to control the release rate, and in turn control cell viability or how much cells die. And we also can use it as a dual delivery system. So we've used it for the metformin and Y15 combination. We are looking to also use it for our minocycline and temozolomide treatment for glioblastoma. So let me end by acknowledging all the help that we get from this for these projects. We are funded by the National Institute of Health. We have a SCORE grant. And then, these my students, UT is an undergraduate institution. I only have had one graduate student, and it was a master's student, and all the rest are undergraduate students. And I always tell the students as well as parents when I do recruitment that these are my workhorses in the lab. And they have a heavy study load and in organization, and we are from one of the lowest financially counties, so the students have to work, so they have outside jobs, but they are still coming to the lab. So I'm very proud of what they've done. As you guys know, this is the last picture we took, in December. We love to eat, right? That's how we socialize with our students. But right now, bottom right, this is the picture that is how our lab meetings and our lab pictures are, right? And the ones in orange are the group of students that work on the project. The rest are actually my high school students from the summer. And I also want to thank Dr. Matirosan's lab from UTRGV for the iron oxide help. And he's also the one who will be helping us with characterization with SCM. And then our collaborator from Arkin Larry from Nowhere Southeastern, who is the one who has got us started with this platinum resistant cancer work. With that, I thank you guys. This is my email. You're welcome to email me for anything. Right now, I am taking questions. Serena, you're going to help me with questions?
So thank you, Swan, for this exciting presentation. So it's very interesting to see how very simple structure can be useful, because everybody is going into complicated structure like 3D printing. So it's very also expensive method for having like results that bring good results for the patients and go to the clinic. So in this way, we use very simple structure. So this is a key advantage of your technology. And but this technology can be improved, and also we can play with the different parameters and reach a good results that is very interesting in this application. So I would like to know if there is a question from the floor first. There are many people here that are interested in your talk, so the room is pretty full for the capacity that we allowed for the COVID-19. Is there any question that from like tissue engineers or drug delivery? So if they are thinking about their question, and I don't see question from the chat, so I can ask myself a question about if you think that this technology can be applied to a different pathology. Or do you think that so the main target is related to brain tumor and other like female tumor, or you can use for
Thank you,
other
Serena.
application?
I definitely think that we can be used, across the board for any application. And in our lab, we are also interested in bone tissue engineering. So that is something we would like to explore next when we try to deliver our microRNAs for bone tissue engineering. And Serena, anything you want to test? Let's work together and we can test something out too. Yeah.
So thank you very much. So if there is no other volunteers for question, then take this opportunity, because now we don't have live opportunities, unfortunately, because of this. And we hope we can have maybe next year, but still we don't know. So if there is no one, we can move to the session. So we really thank Sue-Ann for being here. So you can stay connected
Yes.
if you have time. And if you are connected before logging off from the system, you can ask questions to the next speakers. So otherwise, we say bye to Sue-Ann, and you can still watch
And
the
thank you
conference
so
in the
much
YouTube
for
channel.
everything. I appreciate it.
Okay, so we can go for the next talk. Rach- Rachel Sa, is it correct? How I pronounce it? Yeah. Okay. Thank you for being here. So you can go with your presentation.
Can you see me?
Yes,
Okay.
we can see your presentation.
Hi, my name is Rochelle Dessa, and today I'm going to be talking to you about nitric oxide releasing titanium surfaces for bone integrating orthopedic implants. Titanium implants have been used extensively for dental and orthopedic applications owing to their high mechanical strength and biocompatibility. But there's two major complications that can arise for these type of implants, one due to infection and the other one due to lack of bone integration. Once an infection takes place on a titanium implant, you would have to have repeated revision surgeries, and this can lead to poor implant integration, bone loss, and soft tissue defects. So ideally, to enhance the success of implants, the surface should prevent bacterial adhesion and simultaneously promote osteoblast function. The failure of implants due to infection differs for the type of orthopedic implant. For example, fracture fixation results in a 5% failure rate due to infection. For joint replacement, it's 2%, and for hip and knee replacement surgeries, it's 10 to 14%. Orthopedic infections are mostly caused by the Staphylococcus species, Staphylococcus aureus and Staphylococcus epidermidis, but Pseudomonas is a gram-negative bacteria that is also responsible for some of the orthopedic infections that are quite difficult to treat. The infections can occur through the surgical route or a secondary route from another place in the body where the bacteria has traveled to the site of infection. It is well established that you can prevent these infections from taking place during the first 6 hours of implantation. And this is quite important because what you want to be able to do is try to control an infection before it forms a biofilm, or you want your treatment to be able to kill biofilms. Biofilms are much more difficult to treat than planktonic bacteria. Once a biofilm forms on a surface, it's the adhered bacteria that has this polysaccharide layer that forms around it that essentially acts as a fortress to other bacteria, to antibiotics and any antimicrobial agent that is trying to kill off the infection. Localized delivery of antibiotics as coatings for implants has been seen traditionally as a very strong, promising solution to be able to treat these infections, but given the rise in multi-drug resistance bacteria and antimicrobial resistance, researchers are looking for alternatives to be able to treat infections for medical devices. Researchers have looked at alternative antimicrobials such as silver, zinc, antimicrobial peptides, and nitric oxide. Within our lab specifically, we look at nitric oxide. Nitric oxide is the host's own response to be able to treat infections. Nitric oxide was voted Molecule of the Year in 1992 because it is involved in lots of different functions in the body, from things like cell proliferation to wound repair, immune response, cardiovascular homeostasis, apoptosis, and its antimicrobial function. So what we do within our lab is we look at the response of nitric oxide as an antimicrobial. Nitric oxide is a lipophilic and a hydrophilic gas that can cross the membrane quite easily because it's this molecule that has a very small Stokes radius and it is neutral. And the way it works is that it forms reactive nitrogen species by combining with reactive oxygen species, and these reactive nitrogen species can then damage DNA, proteins, lipids, and inhibit enzyme function. And it's because there's lots of different ways in which the reactive nitrogen species can inactivate the bacteria, that bacteria find it very difficult to develop resistant mechanisms to nitric oxide. Nitric oxide is also good because it's effective against planktonic as well as biofilm bacteria. It's a concentration-dependent mechanism that nitric oxide, so the amount of nitric oxide that is required for its antimicrobial properties is concentration-dependent. At low concentrations, nitric oxide can act as a biofilm dispersal agent, so that if you have another antibiotic or something else in there, it can disperse the biofilm and the secondary agent can kill off the infection. Or at higher concentrations, above 1 micromolar, nitric oxide can be bactericidal. Because nitric oxide is a gas, it is actually quite difficult to deliver, and this limits its clinical utility. Therefore, there's been lots of research around being able to form nitric oxide donors that can effectively deliver this drug to the site of the infection. Within our lab specifically, we work with this molecule called N-diazinium dilates, which form when you have a secondary amine that is a secondary amine nucleophile that couples with a nitric oxide dimer to form this N-diazinium dilate. This N-diazinium dilate can then spontaneously decompose under physiological conditions by protonating this amine to give you 2 molecules of nitric oxide. So you can control the rate of release of the nitric oxide from this donor by varying the acidity coefficient of this amino group. So essentially, the smaller the pKa, the easier it is to protonate, and the faster it will decompose at more acidic pHs. So within our lab, what we look at is lots of different applications for tethering nitric oxide onto various surfaces. And the one I'm specifically going to be talking to you about today is titanium surfaces for sort of dental and hip implants. So the work that I'm going to be describing for you today is based on titanium surfaces that we functionalized with different silane linkers and tethered nitric oxide onto these silane linkers so that we can discourage a bacterial response and encourage an osteoblast response. So the silane linkers we picked for this study were based on some work that had been previously done by a colleague within our department, Jude Kern. She had looked at different silanes onto glass surfaces and seen how amino-terminated silanes can encourage an osteoblast response. And what she found was that this 11-chain silane with an amino-terminated group has a positive interaction, has a positive osteoblast response. So what we did was we took this 11-chain silane, and what we wanted to do is tether it onto titanium instead of model surfaces and see if we could then tether the diazinium dilate onto it and encourage an antimicrobial and an osteoblast response. We also wanted to see how loading it with double the amount of nitric oxide with 2 nitrogens present would affect the antimicrobial and osteoblast response, and we had a control surface with no nitrogen on the surface. Now, of note also is that we have different types of amino groups on these silanes. Over here we have a secondary amino group, and over here we have a primary amino group. So they form 2 different types of diazinium dilates that can release at sort of differing abilities, which I'll go through in a second. So most of this work on this project was done by my PhD student who will be submitting hopefully this week, Miley, within the lab. So we followed the reaction of each step of the reaction, initially using contact angle analysis. What you can see here is that Miley took the titanium surfaces and then she polished them and then immobilized the different silanes on the surface, the AHAP, which has the 2 amino groups, the AUTES, which has just the terminal amino group, and the DTMS, which has no amino group onto the surface. We then exposed the amino functionalized surfaces to nitric oxide gas under high pressure for 3 days and tethered nitric oxide onto the surfaces. So over here, it should tether in 2 possible positions. Over here, 1 position, and over here, it shouldn't tether onto it at all. So this is just a silanization reaction we have observed using contact angle, And what you can see is after we've immobilized the silanes onto the surfaces, we can see that we have an increase in the hydrophobicity of the surfaces, which is because of the alkyl groups of the silane molecule. We then looked at AFM analysis to see what happens in terms of the surface topography, and what we find is that we have an increase in surface roughening from our titanium surfaces after we add the silane groups. And this increase in roughening could be due to multiple layers of the silane that form on the surface, and also over here you can see, for example, the silane sometimes aggregates in clumps onto the surface. We then followed the surface chemistry after silanization and the formation of the diazinium dilate onto the surface. And you can observe that the increase in that the silane has added to the surface by an increase in the nitrogen concentration of the surface, and likewise for the diaxenium dilated surfaces. If we looked at the high resolution XPS spectra, we can also see that this is for the two silane surfaces that are capable of forming the diaxenium dilate, that we confirm the presence of the diaxenium dilate because of the peaks at higher binding energies that are associated with the NO bond that is formed with the diaxenium dilate. We then looked at the NO release profiles by looking at the by using chemiluminescence that quantified NO as it was released off the surfaces, and because it is controlled by pH, what we did was we observed what happened at pH 4, at pH 7.4, and at pH 8.5. So at pH 4, the nitric oxide should release the fastest, and the lower the pKa of the amino group, it should release. It should be an easier release from the surface. So for our three different silanes on the surface, what we can see is that the AUTES group with the AUTES silane with just the terminal amino group releases a burst release of the nitric oxide. At the AHAP, which has two different positions for the nitric oxide, now releases it releases the nitric oxide with a much slower release profile. And the DTMS, which has no nitric oxide, no capability of finding nitric oxide, you can see it has negligible amount of nitric oxide, and nitric oxide you see is just fizzy sorbed. And titanium is the other surface in there, and you can also see that there's no nitric oxide or just fizzy sorbed nitric oxide adhered onto the surface. When we look at the surfaces at pH 7.4, which is physiological pH, it follows a similar trend. The AHAP surfaces release slower than the AUTES surfaces, and you see some fizzy-sorbed nitric oxide on the DTMS surfaces. And when you move to pH 8.5, what you also notice is that the release profiles are once again longer because of the higher pH. One more thing to consider, which complicates the situation a little bit more, is that for secondary diaxenium dilates, you always get 2 moles of nitric oxide release. But in the case of primary diaxenium dilates, the amount of nitric oxide release is actually based on the pH. At acidic pHs, you get all nitric oxide. At pHs between 5 and 8, you actually get a mixture of HNO and NO, and at high pHs, you get just HNO. So this is why people mostly use secondary diaxenium dilates, but in this case, we wanted to test whether this osteogenic linker could be used as a tether to produce primary diaxenium dilates that can also function as an antimicrobial tether. So what we thought was going to be quite an easy study initially actually turned out to be quite complicated. And we did a lot of mechanistic studies, which I won't go through today, but it's just recently published in ACS Applied Materials and Interfaces this year, into how the pKa, the class of the amine, and the quality and the type of the silane layer actually affects the rate of release of nitric oxide from these surfaces. So it's not a simple, we can do one thing and we get one result. It has to be varied on all these 3 different conditions that have to play into one another to be able to get a controlled nitric oxide release. But what we found when we looked at the biological response is that we looked at Staphylococcus aureus and we looked at Pseudomonas aeruginosa, which are the 2 bacteria, 1 gram positive and 1 gram negative, that are responsible for most orthopedic infections, is that the surfaces are capable of releasing nitric oxide in a concentration that is bactericidal. So if we look at the SEM micrographs of how Staphylococcus aureus responds to these surfaces, what we can see is that after 6 hours on the titanium surfaces, you can see the biofilm formation as well as bacteria adhesion to the surface with and without nitric oxide on the surface. And the DTMS, which is our control surfaces, also has bacterial adhesion and biofilm formation on both these surfaces. For the AHAP and AUTES surfaces, you can see that before nitric oxide has been tethered onto the surface, you see lots of bacteria adhered, and after the nitric oxide has been attached to, has been immobilized onto the surfaces, you can now see that we have in fact been able to kill bacteria Staphylococcus aureus on the surface. The same trend is also observed for the gram-negative bacteria Pseudomonas aeruginosa, where both our nitric oxide silanes have been able to effectively kill off the bacteria. So if we look at the biofilm CFU response, so we look at numbers specifically of what happens. We've looked at Staphylococcus aureus and Pseudomonas once again, and we've looked at it after 6 hours and after 24 hours of adhesion. So what we find is that for our surfaces with no capability of nitric oxide, we don't see a decrease in bacterial response as expected. But for our our surfaces with the aminosilane that have the ability to tether nitric oxide, we see, for example, the AHAP surface, we see that there is a 1.5 log reduction in the amount of Staphylococcus aureus after 6 hours compared to the silanized surface. And we see that the AUTES surface has a 0.8 log reduction in the amount of bacteria after we tether nitric oxide onto the surface at 6 hours. Now, after 24 hours, actually the same, even though the number of bacteria grow, the same trend holds. We see a 1.5 log reduction here for the AHAP surface and a 0.8 log reduction here for the AUTES surface. And just to remind you, the AHAP surface, which shows the better bacterial response, has the 2 amines that are capable of binding nitric oxide. The AUTES surface has the 1 amino group capable. So this should be able to hold a bit more of the nitric oxide onto the surface. When we look at Pseudomonas aeruginosa now, what we find is that Pseudomonas aeruginosa is notoriously more difficult to kill than Staphylococcus aureus, but we find that the same trend exists. After 6 hours, the AHAP surface kills 0.8. It has a 0.8 log reduction in the amount of bacteria, and the AUTES surface also has a 0.8 log reduction in the amount of bacteria for the nitric oxide surface compared to the silanized surface. When we look at the response at 24 hours, for the AHAP surface, we see a slight reduction. It's a just a 0.3 log reduction because the bacteria have grown after 24 hours. But on the AUTES surface now, we see that there is no difference in response. The bacteria for the AUTES surface has grown completely. So we wanted to then see what happens in terms of a biological response. So we then cultured human primary osteoblast cells onto the surfaces to see what happens. Do we still get a positive cellular response on the surfaces? On our titanium samples, the osteoblasts, like the titanium samples, they grow quite well with or without the nitric oxide on the surfaces. And on our DTMS surface, likewise, with no ability to form the diazine dilate donor, we see that the nitric oxide or form surfaces that are capable of releasing nitric oxide, we see that there's the cells grow quite well. Now, on our 2 silanized surfaces where they have the capability of tethering nitric oxide, what we find is that our AHAP surface, which has the higher amount of nitric oxide, the slightly better bactericidal effect, they don't like the silane surface, the osteoblast, but on the nitric oxide surfaces, the cells do grow a little bit better, although the morphology is, they've got slightly shrunken morphology. Whereas on the AUTES surface, as we had previously seen, the cells do like these surfaces and they are displaying a bit of a positive response to the surface, and they have the cobblestone morphology that is indicative of osteoblast function. So we are getting a positive cell response on this type of surface, which has a bactericidal capability, not as much as this type of surface, but the results are promising. So what we've been able to develop in this regard is surfaces that are capable of having an antimicrobial as well as osteoblast function. The prevention of bacterial adhesion and biofilm formation on surfaces while maintaining osseointegration is a significant medical challenge. We have just started a little bit into this direction, and we've developed surfaces that are perhaps capable of achieving both of these functions. We've carried out extensive, comprehensive mechanistic analysis for the nitric oxide release, and we have found 2 surfaces that are capable of having that dual function. And currently what we're trying to do in the lab is trying to look at the situation together. We're trying to culture the bacteria and the mammalian cells together to see what happens in a sort of race for the surface phenomenon. And with that, I would just like to acknowledge my group, without which none of this research could be done, and as well as our funders. And most of this work, as I said, has been carried out by Miley here. And thank you for your attention and your time.
Okay, thank you very much for your presentation. Is there any question here? Maybe Nihal, do you have a question for the speaker?
Hi, Ashar. Thank you very much for the interesting talk. My question would be, what would your opinion about the length of the silence you can go up? And would that have a positive or a negative effect?
My colleague, Jude Curran, had done a lot of this preliminary work based on the length of the silane, and what she found was that the 11 chain length silane actually had the most positive cellular response in terms of osteoblast cells. And when she used smaller chain silanes, for example, if the alkyl chain length was only 3, it didn't have that response. So the osteoblast response depended on the length of the silane, the stiffness of the layer, and the surface chemistry at the end as well.
Okay. Interesting. Thank you. You are muted, Serena.
I'm muted. Oh, sorry. Yes. So I was asking a little question from my side. So how do you think your technology can be applied to orthopedic implants? And if you think the shape of the implant is suitable, like this 3D shape, so to be easily modified on the surface with your technology?
So what we've also started looking at is we've started to additively manufacture some of these titanium implants into 3D structures to see whether we can get the coatings on a 3D type structure, because if we get too thick a coating, it could, for example, block some of the pores of a 3D type structure. We are getting a thin enough monolayer that it is allowing cell infiltration and maintenance of the 3D structure with the porosity and everything, with the coatings intact so far for some of our studies that we haven't published yet on 3D structures.
Okay. Thank you very much.
Thank you.
So if there is no question, we move to the next presentation, which is from Dr. Mariniello, Maria Donatella, and she will talk about breast implant.
Thank you.
Devi fare aspetta. Così piccolo, si vede. Ok. Capito? Alzo di sì, condividi. Ok. It's coming? No. Oh. Okay. Okay.
Good afternoon, and thanks to Professor Danti for our invitation. Today we'll talk about how to improve outcomes in breast reconstruction from the current techniques towards tissue regeneration. And we all know that breast cancer is the most frequent cancer among women worldwide, and considering that the growing number of women live with a previous diagnosis of breast cancer, today for the surgeon, aesthetic outcome is a fundamental goal as well as the oncologic control. And as a consequence, the surgical treatment has radically changed over time. And we started with radical mastectomy that was very mutilating for the patient, and then passed through breast conserving surgery, pioneered by Professor Veronesi, that was less invasive than mastectomy, undoubtedly, but not always associated with good aesthetic outcomes. And so we arrived in the 90s to the oncoplastic approach that combines principle of plastic surgery and oncologic surgery, and whose aim is to optimize the aesthetic outcome by removing the breast lesion and contemporarily reshaping the surrounding gland in cases of breast conserving surgery, or reconstructing the new breast in case of mastectomy. And currently OPS represents the gold treatment strategy. And moreover, the importance of aesthetic outcome has been further stressed in the last decades due to the so-called Angelina Jolie effect, because this well-known actress has drawn attention to hereditary breast cancer. And so more and more women that are carriers of this mutation require a prophylactic surgery, that is surgery on currently healthy patients. And in this setting, the aesthetic outcome is of paramount importance. And so surgery has evolved in order to get as close as possible to this ideal of breast. And so the currently available techniques for breast reconstruction can be divided into autologous tissues as based with the patient's own tissue, which is moved from a donor site, such as the abdomen or the thorax, to the defect site, or implant-based using tissue expanders or prostheses. The autologous reconstruction has some advantages because it is more natural in shape, texture, more durable in time, but it is more technically challenging, needs longer learning curve, is time consuming, longer hospital stay, and has morbidity on both the donor and the recipient side. And so by far the most commonly used breast reconstructive method is the implant-based. This reconstructive technique can be carried out either as a single-stage procedure direct to a permanent implant, or a two-stage procedure by implanting a tissue expander that has inflated over time and then replaced some months later with a permanent implant. And because traditionally used tissue expanders require multiple injection of saline solution through skin, that can be painful, that can increase the risk of infection, recently the Aeroform device has been introduced, that is a remote-controlled needle-free tissue expander that releases CO2 from an internal reservoir, so that the patient can have a daily gradual expansion made by herself. And traditionally, these devices were implanted only in two positions, completely under the pectoralis major or in a dual plane position. So with the upper pole covered by the muscle and the lower pole covered by the inferior mastectomy skin flap. But more recently, another technique has evolved, that is the prepectoral implant. And it is a muscle sparing and minimal invasive technique that leaves the pectoralis major muscle completely untouched. And the currently used implants can be divided depending on their shape and their surface texture or filling, because they're all made of a shell that gives strength and a filler that gives bulk and consistency. And concerning the shell, the surface texture of the shell can be smooth or textured. Smooth implants have the drawbacks that they move within the breast implant pockets, while texturization increases the stability of the device, but is associated with an important complication that we will see later. While concerning the filling, it can be saline or silicone, but saline prostheses have a consistency that is like that of water, and they tend to wrinkle under the skin. while silicone breast implants more accurately mimic the breast feel, and so they are by far the most commonly used. And these silicone implants have evolved into different generations based on the shell thickness and gel viscosity, because the first generation used in the 60s and 70s were characterized by the thickest shell and more viscous gel, and they felt too firm compared to natural breast, and they were associated with a high incidence of capsular contracture. So the second generation was developed that was characterized by reduced shell thickness and gel viscosity. But because of this thin impermeable shell and less viscous gel, they were associated with a high rate of shell rupture and gel bleed into the surrounding tissue. So the third and fourth generation developed, designed under FDA criteria and improved quality control with wider implant shape and surface textures, till the fifth generation that is currently used, that is represented by anatomically shaped textured implants with a highly cohesive gel that are able to maintain their shape even if the shell is broken. And the advantage of this technique are that it is very simple, very quick, and very satisfying in terms both of cost and aesthetic outcome, but it is associated with some disadvantages that are very important. And first of all, capsular contracture and lanaplastic large cell lymphoma. Capsular contracture is the most common complications of implant-based technique because it is characterized by a foreign body reaction against the implant, leading to scar tissue formation, pain, deformity, and needing surgery to be corrected. While anaplastic large cell lymphoma has been found recently by the FDA to be associated with the texturization of the implant, and it is a rare form of non-Hodgkin lymphoma that presents an average of 7-10 years after the implantation as a large peri-implant seroma containing malignant cells. And it requires surgery to be corrected and chemotherapy in cases of advanced diseases. And for both of these important complications, the most credited theory is represented by the biofilm hypothesis. So it proposes that it is an endogenous flora and bacteria that gain access to the prosthesis during implant, attach to it and form this biofilm, and leading to a subclinical infection. But when the number of the bacteria reach a critical point, it triggers a chronic inflammatory process, leading to scar tissue formations and so capsular contracture, and in genetically susceptible patients to a T-cell dysplasia and so to the lymphoma. And other techniques have been recently introduced to enhance the possibility of breast reconstruction for our breast cancer patients. And one of these is autologous fat grafting. The advantages of this technique is that it is very inexpensive, simple compared to alternatives, and is very successful in correcting small contour deformities or treating damaged irradiated tissues. But it has two main drawbacks. So the unpredictability of outcome and the graft volume loss of around 30%. So it is possible to reconstruct a whole breast with only autologous fat grafting, but it requires multiple sessions with unpredictable outcomes, and it is optimal for small to medium breasts, but not for larger ones. So considering all the aforementioned drawbacks of the currently available techniques, the question is it possible to obtain the same results without the same problems? And with this in mind, it is obvious that a further step forward is now necessary in breast reconstruction, and it is represented by tissue engineering that aims to restore form and function of breast by regenerating new breast tissue. And the possibility to be successful depends on the capability to find the correct blend of cell sources, microenvironment, and scaffolds. And considering cell sources for tissue engineering purposes, in the breast reconstruction, even if there is no consensus on the ideal cell source, stem cells are the preferred because of their high capability of expansion. Autologous stem cells are the preferred because of the lack of immunogenicity. And adipose-derived stem cells in particular are rapidly becoming the gold standard because they can be easily obtained through liposuction, and they are able to regenerate new adipose breast tissue. And considering the currently published literature, two approaches can be seen in using this kind of cells. The first is to isolate and culture pre-adipocyte prior to implantation, and the other is to use growth factors to recruit resident pre-adipocytes. For example, cell-assisted lipotransfer that has been introduced in 2006 requires to enrich the autologous lipoaspirate with adipose-derived stem cells that have been previously harvested and expanded ex vivo in order to reduce the graft volume loss. And cells are important, but not sufficient to recreate the breast tissue because they need the environment and they need the scaffold for biological and structural support. And for tissue engineering purposes in breast reconstruction, these scaffolds should be biodegradable, versatile as regard to manufacturing methods, suitable for sterilization, have optimal micropores, and they have to be biomimetic with the suspensory ligaments because they have to recreate the mechanical characteristics of the suspensory ligaments that are the main components giving support to the breast. And they should have an appropriate mechanical strength. So they have to be flexible enough to be comfortable for the patient and to allow tissue and growth, but should be able to keep their shape despite handling during surgery and despite the mechanical forces acting on the device itself due to the patient's daily life activities. And the currently studied scaffold can be divided by structure into solid or hydrogels and origin. And each of these is some pros and cons because natural scaffolds are characterized by low immunogenicity, but they have too rapid degradation and limited mechanical strength, while synthetic scaffolds are editable in their characteristics, can be added with ECM components, growth factors, so they're very versatile for use, but they can release some harm byproducts and so can determine inflammation or pH alterations. And finally, the biological scaffolds biomaterial that are decellularized extracellular matrices that are characterized by minimal inflammatory response and very accurate mimic the native extracellular matrix tissues are bidimensional and they have a very high cost. So even if basic research has rapidly evolved during the last decades, the translation from laboratory to clinical reality still remains a huge challenge. Because apart from being able to engineer durable and functional tissues that accurately mimic the natural breast, one must also navigate a complex regulatory process. Concerns regard storage, transportation, possibility of contamination. It is of interest that these products do not easily conform to either of the FDA classification and biologics or devices. And first and foremost, patient safety needs still to be assessed because in the development of any new treatment strategy in medicine, patient safety is a primary consideration. And the problem with these biomaterials might be that they might determine cancer regrowth or hinder cancer detection. And indeed, scaffolds, the presence of the scaffolds might reduce the sensitivity of the manual breast examination or could hinder cancer regrowth within its boundaries. But also surgical considerations are important in the setting because the site of positioning of this biomaterial must be defined yet, because probably the optimal site of positioning would be under the pectoralis major, because in this case, the biomaterial would be separated from the gland in which the cancer originates through a layer of muscle. And this would allow also a better vascularization because muscular vascularization is higher than glandular vascularization alone. But on the other hand, it is to be considered that subpectoral positioning is not so minimally invasive. So it could be uncomfortable for the patient and might pose an excessive mechanical force on the device itself. So also these considerations must be done before using this biomaterial. And so to conclude, even if the use of tissue-engineered biomaterials has the potential to revolutionize clinical practice in breast reconstruction, many have yet to be convinced of this potential. And critical considerations are ethical, regulatory, financial, and first of all, clinical, because most of all, we must be sure prior of using this biomaterial in patients that we are able to recreate large volume constructs that do not interfere with cancer detection or do not promote cancer regrowth. And it is only close multidisciplinary cooperations between scientists and clinicians and industry that can move this from potential to reality. So thanks for the attention.
Thank you very much, Donatella, for this. I'm okay. Okay, for this interesting talk. So, which is more very clinical oriented. So for us as engineers, it's very important to have the clinical outlook when we are developing our materials. So, is there any question that is coming from here? So I can ask you how you see the next step in this tissue engineering. Do you think for this kind of application, we are gonna go towards bioprinting, so printing cells and materials together, or for other types like implanting and materials? What is the next future that you see in front of you?
Thanks for the question. And in my opinion, the future is represented by biomaterials that can be enriched with autologous cells that have been harvested prior by the patient and expanded ex vivo, because the problem of enriching the biomaterials with cells is the immunogenicity and the difficulty of culturing cells in labs. And so while isolating adipose-derived stem cells from lipoaspirate is very simple, and it is characterized by the absence of immunogenicity because autologous cells. And so being able to have a biomaterial that is biodegradable with all the characteristics that I have said before, that can be enriched with autologous cells would be, in my opinion, the future, because it would be simple, completely biocompatible, and easy to obtain compared to alternatives.
Okay, thank you very much. So I think we can move to the next speaker. I'm going to introduce Nihal Indindrana from Sparta Medical, and so he will give a different talk, I guess, from the previous before. That is, in any case, is very relevant. So for researchers and engineers and medical doctors. Thank you very much.
Thank you very much, Serena, and thank you for the invitation. Do you see my screen?
Not yet, but maybe it's coming. Yes, it's coming.
It's on? Can you hear me? Serena, can you hear me?
Yes.
Okay. So, good afternoon, everybody. I would like to thank the organizers for the event. I was going to come up until Sunday, but my flights were canceled one time too many. I will try to tell you about the translation of the research results towards applications, medical and other. And Sparta Medical came from an INSERM unit from France, Strasbourg. And what we are doing is supramolecular coatings for medtech and other sectors which have multiple functions. So we see ourselves as a customized coating company, and we are trying to customize the surfaces, for example, for the personalization of implants. And we develop nano microscale coatings which have several applications at the same time and which can prevent complications. It can be antimicrobial, antiviral, or anti-inflammatory. These are applied by dipping or spraying robots, and they can conform to any kind of surface. And in the end, due to the inherent properties of the coating, we can have different effects. Here you can see how we can kill bacteria in the context of orthopedic implants as covered before. And the problem here is that for nearly all implants, there's a certain risk of infection, as Rachel showed. But there is another aspect. When you look at the hospital-acquired infections, the nosocomial infections, which touches about 5% of all hospitalized patients, about 50 to 70% of these are related to the presence of a medical device. And when this happens, there's an additional cost of about 10,000 euros per each patient. And the worst case is that even if you solve this initial problem, the next time you have the implant, you make a revision surgery, you have an infection rate that doubles. So for this, our solution was to develop an antimicrobial coating that can conform to any kind of implant. And in there, what we were looking for is that from a value proposition point of view, is to have a new antimicrobial agent with a large spectrum of activity, and most importantly, due to the presence of the antimicrobial resistance, to have something that the bacteria cannot develop resistance to. And the second part is to have simultaneous activity. It is not just antimicrobial, it is antimicrobial, antiviral, and anti-inflammatory at the same time. Antiviral is not that important for the most implant context, but anti-inflammatory is quite important because, as shown for the breast implants, the immune reaction to the biomaterials is an important part of complications. So another important part about the technology is to be able to apply to any kind of material and to be able to industrialize it either in the sense of robots or sprays without having any chemical treatment. So how this started? That is the part of the story I wanted to tell you about, and I think you might find interesting. This project comes from University of Strasbourg, and it originates from a European project called Imogel, and continued in another European project called Pambiora. And the first intellectual property came during the projects, which was further matured by the local technology transfer office and led to other patents. And in the end, with the technology transfer office, we decided that the best way to valorize these results is to create a company. So we started an incubation supported by the Grand Est region in France, which we are located. And then we won a national competition, an innovation competition, which was the starting point of the company. And at the moment, we are a team of 6, and the company was created about 9 months ago. And in the original project, the part of antimicrobial effect was a afterthought, actually. And what we were looking at is to decrease the complications related to the chronic inflammation. Because one of the problems with biomaterials, as our body generally treats them as either inert material or things that it cannot degrade, the resolution of inflammation around the implants is always problematic. Either there is a strong inflammatory effect which leads to chronic inflammation, and in some cases requires additional surgeries, or in the case of not full repair, it can lead to encapsulation of fibrosis and again limited function of the implant. So the hypothesis of this project is that if instead of trying to control just the surface of the biomaterial, if we can incorporate the patient's own macrophages in a given phenotype, we can create a microenvironment around the implants, which will be beneficial for the healing, so we can contribute to the resolution of the inflammation. And in the end, by having this specific microenvironment, the implant heals and integrates instead of moving towards chronic inflammation. So the project had many aspects. One part is about immunoprofiling, which we still work on. And the other part is having three different kind of structures. One is where we can actually encapsulate these cells, and the other providing patterns, just like in the case of breast implants, so that we can control the incoming cells. And the third part was an adhesive that can apply this structure onto the surface of the implant, no matter which implant. And for this, we were using bioprinting. And when we were looking at the potential adhesives, we were looking at layer by layer structures. And in these structures, we were looking at how we can cross-link them, whether we can have self-cross-linking structures with aldehyde groups, and so that we can attach these hydrogels. This was one of the formulations which is based on PLL and HA aldehyde. And one thing we have started to notice is that we were loading anti-inflammatory molecules inside these to have effect on the macrophages. These are the cytokine secretions by primary macrophages in the presence of these coatings. And once we add interleukin-4, which is an anti-inflammatory cytokine, what kind of reaction we get. But we started to notice that the nature of the coating has an effect by itself. If you look at the TNF-alpha levels or interleukin-1 beta levels, just having a self-cross-linking system with HA aldehyde has an effect. Having IL-4 accentuates this effect, but there's an effect by itself. And while we were looking at different configurations, we have come across one configuration which has the same anti-inflammatory effect, but at the same time, at a very narrow molecular rate, it also have an antimicrobial effect. And this was a very good case for us, because since the beginning of the project and during the review process of the project, this was the main question. If you modulate the immune reaction too much around the implant, what will happen? If there is an infection, how we are going to deal with it? And now we have a coating which is dealing with these two problems, which looks like mutually exclusive simultaneously. So this was the basis of our first patent application. And this is how it works. For example, this is an SRS growth on a normal implant surface. And with our coatings, we are basically popping up these bacteria. It is effective immediately, and with an efficacy of around 99%, and no growth of bacteria. And then when we try this in vivo with an infection model in rabbits with a hernia mesh, we also have prevention of the infection, and also thinner fibrous capsule compared to the implants, which is an indirect indicator of the anti-inflammatory effect. And as mentioned, there are several parts to turn an idea from an idea to a product. And one part, you need to have the proof, both in vitro and in vivo. And after that, there are a certain set of experiments that needs to be done for showing the biocompatibility of your system. It doesn't necessarily mean that you take several biocompatible materials and put them together and it will stay still biocompatible. Even if that is the case, it needs to be proven. And also, you need to validate all your claims. For example, I talked about absence of bacterial resistance. There is a standard for that, and you need to show it with that. And then more logistic and technical questions come into play. How long you can store your coating on the implant? For example, when we talk with implant companies, minimum is 2 years, preferably 5 years. So you need to show that. you need to show that you can apply it to different materials and have results with it. And also, you need to show that it will be suitable with the sterilization method, not just one sterilization method, but the sterilization method used by different segments in the industry. And at that point, you need to look at, okay, you start it with a more general solution, but then you need to look at what is the real target, where is the real pain point, and where you can first get into an actual application. So what we are looking at is an intersection of the antimicrobial coatings, which is a more general market, and the medical devices. And when you look at that, for example, at the moment, the biggest need is with the diabetic ulcers, where the infection rate is around 40%, and also hernia meshes, which has a 7 to 8% infection rates. And with the with the current COVID situation, one other problem is that we started to get a lot of demand for having something that also have antiviral activity, not for the medical context, but for more general context. As you can see with the different applications, the needs are quite separate. And at the moment, what we are offering to the medical device companies is to develop coatings which are product specific, optimized for the specific microenvironment of the product, specific mechanical and cellular environment of the product. And then once it is validated, we can sub-license it or become their subcontractor for applying the coating in our facilities. And the second part we are working on is to have a generic coating kit which can be used directly in the operation theater for such as trauma surgeries. And in this context, we have 3 products in development: an antimicrobial film, a hydrogel, and an antivirus spray. And with the current situation, and this is the part I wanted to emphasize, we are coming from a health institute. We have a very medical focus, but with the current situation, the medical concerns kind of permeated into all aspects of life. So we started to get demands from metros, demands from airports, demands from high-tech companies to have something that can clean the surfaces. And in this context, we have developed a formulation which is effective against SARS-CoV-2. And what we are trying to do now is to see how we can have customized coatings for different conditions, and at the same time continue our product development for the medical device segment. And this works with initial feasibility study with the company, and then developing the coating with them, making the necessary functional tests depending on the application area, and resulting in a transfer of the technology for the specific implant. Because one part about the regulation is that you cannot have a general regulation for a coating. It is separate the assess for each kind of implant. So this is an important point for each company. So this is a risk for them. And so it is much better to develop a coating together rather than trying to get something off the shelf and try to apply to their product. And currently, we are working on this further in an ongoing project, which also takes into account the personalization by means of immunoprofiling, and actually a development of an instrument that can quantify how a specific patient will react to a specific set of implants, so that you can develop the necessary coating in that context. This was a result of a EU project with the first patent, which led to another 3 patents in the development and 7 scientific publications. And recently, we have obtained one Deep Tech award from the French Bank of Investment, and our activity around the anti-COVID sprays has been widely covered in French media, and we hope to develop a product next year on this application. Also, I still try to stay connected with the science part. So these are some recent activities we are looking at. What we do is for me the interface engineering between the cells and materials, be it bacteria, immune cells, or the cells of a specific type. So we are keenly following all the developments in these areas. So with that, I would like to thank our founders, the structures that support us, particularly SEMIA, SatConnectus, and the Grand Est region, and my team, and our academic partner, INSAM Unit 1121. Thank you for your attention.
Thank you very much, Nihal. Maybe Rachel has a question as she works in the similar field. Do you have any question from Nihal?
You're saying that the coatings, how long do they have to last on the shelf for when you develop these coatings? What do companies expect for your clients?
Depends on the application. For example, for catheters, it is about 3 days to 5 days. For the orthopedic implants, it can be up to a year, because there are cases where the infection happens after a few weeks, but at least few weeks, 3 to 4 weeks. And for dental implants, these secondary infections is very common. So for them, they are talking about years, but years is like a holy grail. Like at the moment, there is no real coating that can stay years in place and still be active, because it is a bit counterintuitive also. Like it needs to be integrated how it is going to work. But I mean, realistically, it is within the range of a month.
Okay. And what sterilization methods are most commonly used
It
for
is
some of
based on
these
two
coatings
things:
within?
either the base material or the habits of the company. The most common one is gamma ray sterilization that I have seen. Ethylene oxide was very common, but it is going to go away slowly because there is remnants. Autoclave is not as common in the industry as we use it in the lab. So it is used, but like radiation sterilization is much more convenient for industrial scale.
Thank you very much. Thank you, everybody. If there is no question, I think we can go for the coffee break. So I kindly invite you to log off from the system, so we allow more speakers for the next session. And we can hopefully see, Nihal, we can hopefully see next time.
Yeah.
Very unlucky that they cancel your flight.
Bye-bye.
Yeah. Okay, so thank you for being here anyway. Thank you. Okay. Bye-bye. See you later then. So in half an hour, we will start again. Okay. Bye. Good afternoon to everybody. So we are back from the coffee break. Can you hear me? Yes. So and then we are gonna start with the last session of today. That is the first day of the conference. So the last session will start with the presentation of Dr. Laura Stolfi from University of Padova. I'm very glad to introduce her. She's my friend and colleague, and we have worked together for a long time with different projects. So please, Laura, you can have the speech.
Thank you, Serena. And I'd like to thank you and also the organizing committee, because even if the COVID-19 give us the opportunity to follow this really interesting workshop. So, are you able to see my presentation? Okay, so I will start. Today, I will talk to you about the biocompatibility tests that were made in my laboratory on nanoparticles useful to develop inner ear therapies. Why the inner ear? Because we are interested on the sensory neuronal hearing loss, and this region, the inner ear, is the site where it happened, the acoustic transduction. Indeed, the amplified acoustic wave is transmitted from the eardrum through the ossicle to the oval window on the cochlea, so in the inner ear. The cochlea is a spiralized and compartmentalized structure where the acoustic wave stimulates the basilar membrane, because it moves the basilar membrane, leading the hair cells, so the sensory cells, to transduct the mechanical stimuli in a neural activation. So the patient that they lost the hearing because they lost this sensory cell, the only way to recover the hearing is use the cochlear implant. The cochlear implant is an electronic high technology device able to transform this acoustic wave in electric stimuli, bypassing the sensory tissue. So it directly stimulates the acoustic nerve. Which are the risks and limitations for the people that underwent the cochlear implant surgery and then they wear the cochlear implant? They could lose the residual hearing and the tonotopy. And this leads to incomplete language understanding, and usually they have problem with the music perception. Another problem is that the implant could interact with the static electricity and the magnetic field. So this patient could have problem if they need a diagnosis by the MRI, or if they want to use a mobile phone, or if they want to pass the metal detector, or apply another hearing aids. Secondly, another problem it is that the cochlear implant has battery charge dependency, and the last but not least problem is the aesthetic problem. So it could affect also the quality of life of this patient. So the question is electric biocompatible nanofiber could be employed to mimic the function of the basilar membrane? Because this material, the piezoelectric material, have this reversible effect. If it is subjected to mechanical stress, it could generate a voltage. And in the opposite, if we applied a voltage to this material, it changed its shape. So it's similar to the basilar membrane. So our idea, the possible idea is put this material under the basilar membrane. And so we will obtain a new implant that is more simple, because we have not all the complex electronic implant. It is self-powered, it is less invasive, waterproof, and because it isn't ferromagnetic, there are not interference with the electromagnetic fields. The idea to use the piezoelectric material for the cochlear implant starts since the 2000, and it was published the first paper that speak about this possibility. And since the 2000, a lot of researchers try to use this material and to improve the material to have the opportunity to perform, to product a new implant that is able to activate the neuron bypassing the hair cell. Our project, the project in which we were involved, have these different aims. And firstly, we decided to apply these nanotechnology strategies to overcome the drawbacks of piezoelectric cochlear implants. Because the use of piezoelectric nanoparticles, for example, use the polyvinylidene fluoride nanocomposite, could improve the material sensitivity. And at the same time, we use a micro nano fabrication that can help us to improve the interface between the piezoelectric material and the neuronal tissue. Today, I'd like to show you the results that we obtained testing the biocompatibility of different nanomaterials that we decide to use to combine with the PVDF material. So I will speak about the barium titanate and the lithium niobate that are piezoelectric and then increase the piezoelectric effect of the PVDF. And then I would like to speak about the glycerol monoamide nanoparticles that we used as a drug delivery system. We tested all this material on the OCK cell, that are cells mimic of the hair cell, and so the sensory tissue. And also, we tested the same on two different neuronal cell lines. But today, I'd like to show you the results about the hair cell, because I'd like to speak about how to preserve the residual hearing. The biocompatibility tests that we made were analysis of the cell adhesion, looking at the shape of the cell, toxic analysis by cell viability assay, in order to understand which is the maximum amount of nanoparticles that we can combine to our PVDF material. And then we decided also to analyze the oxidative stress and the inflammation in order to be sure that there are not distress of the cells. About the barium titanate. The cell viability assay show that it is never toxic after 22 hours. And about the cell adhesion, so the morphology, after 72 hours at the highest amount that we tested, we found that the cytoskeleton is still well organized. The cell have the typical polygonal shape, and also the nucleus are rounded with good diffusion of the chromatin. So they are in healthy condition. To deeply understand if there are some cell distress, we analyzed the ROS production, and so we didn't find oxidative stress. And we analyzed also the expression of the cytochrome C. Cytochrome C is a marker of cell distress. When the cells are not in healthy condition, the membrane of the mitochondria are damaged, and the cytochrome C goes outside of this mitochondria in the cytosol. As we can see, after 72 hours at the highest amount of nanoparticle tested, we have spotty expression of the cytochrome C. So it stays inside of the mitochondria. So again, there are no distress because they are in healthy condition. About the lithium-ion battery. The serviability again shows that there are no toxicity at all the doses and time tested, and as reported in literature, also in OCK3 cell, the lithium-ion battery increase the cell proliferation. About the cell adhesion, so the morphology, again, we have a good organization of the cytoskeleton and a healthy condition of the nucleus. Oxidative stress wasn't detected, and the same, the cytochrome C stayed inside of the mitochondria. So both lithium-ion battery and barium titanate are good and biocompatible nanoparticles that can be applied to the PVDF. Another issue of the cochlear implant is the impedance of the electrode, because when we insert the electrode in the scala tympani, because it's a foreign body, we have the foreign body reaction. So we have a tissue grow around the electrode. This is an example of an experiment in which we put a silicone rod inside of the scala tympani linear peak, and as you can see, after 2 months, we have all this amount of tissue grow around, and so we lose the possibility to stimulate the basilar membrane. To avoid this, we thought that we need to apply a drug delivery system. As drug delivery system, we used the glycerol monoelate nanoparticles. These nanoparticles are toxic at the increasing amount of nanoparticles after 50 microgram ml of amount. It isn't this death dependent on the time, it is only dose dependent. And if we look at the morphology, with the 25 microgram of ml, we have a good condition of the cell because the shape stays polygonal and the nucleus are in a good condition. When we test the 50 microgram ml of nanoparticle, you can see that we have thinner shape, so the cells start to be stressed, and we have a lot of microvesicle, maybe lipid vesicle inside of the cell. Because it's a delivery system, we decided to combine at the glycerol monoelate the dexamethasone. Dexamethasone is an anti-inflammatory drug, and we have already tested this compound in guinea pig, and we saw that the slow release of dexamethasone added in low amount to cochlear implants is able to reduce the hearing loss secondary to the insertion trauma. Indeed, is able to reduce the tissue growth inside the scala tympani. And our drug delivery, glycerol monoelate nanoparticle, combined to dexamethasone, are able to release dexamethasone along the time, so we have a slow releasing of the time, and it works because after 48 hours we can see that the amount here in green of dexamethasone entrapped, so close inside of the cell, is increasing. Now, the next step of the project will be try the in vivo biocompatibility of this new material. So we think that we will use the rat as an animal model. We want to use the ventral surgery approach by cochleostomy to insert the material under the basilar membrane. And then we will test the auditory function by the auditory brain response analysis and look at the foreign body reaction by histology assay. I have to thanks all the people that work in this project, that are the postdoctoral Erika Gentilin and Eddie Simoni, and the PhD student that works, Maria Rita Candido, that works in my laboratory. And also I have to remember Filippo Valente, that he was a PhD student in my laboratory, and he worked on the glycerol monoamylase assay. And then I have to thank Serena Danti and Professor Stefano Berettini that involved us in this project. The project was founded by, at the beginning, the Italian health minister, and now we have tested all the nanoparticles also in the 4 nano eardrum project, that is an European project in which we are again involved. Many thanks for your attention. If you have some questions, I'm here.
Laura, thank you very much. I think your presentation is interesting, as you show that for the ear application, nanoparticles can have several functions, like giving electromechanical stimulation, but also release drugs. And the main problem is in this organ is the high cytotoxicity that some components can have. So it's very important in this application to be very careful. Otherwise, we can have like a loss of hearing function, no? So how do you think these nanoparticles can be delivered into the inner ear, so in order to provide the therapy? How do you think they are injected, or you are released from like a gel, or in which way you think we could apply the nanoparticles?
Okay. I think that the best things will be directly apply this material to the basilar membrane. But it is not so easy to direct this application inside of the cochlea, because the cochlea is very, very little and it is not so easy to arrive at the site. So maybe some hydrogel or some other kind of device of application could be help us to put this cell inside, to put this material inside. The nanoparticles that I show, I think that will be used simply coating the PVDF material. And then insert this material in the cochlea. Serena?
Can you hear me? Yes.
Now?
So is there any question from the auditorium to her? So no. Laura, do you want to add anything to your answer or not? Because maybe I interrupted you, I don't know.
No, it's okay.
We have some delay, yeah, okay. Okay, thank you, Laura. Thank you again for being here. You were very present this morning, so it was nice. So please, if you like, continue to watch the conference in the YouTube channel. Thank you.
Thank you.
So now we are moving to the final presentation for today. So hi, Wim Demalske. I'm not sure if I pronounce your name correctly.
Yeah,
maybe not.
It's
Yes?
Wim the
More or
Malske.
less. Okay.
Thank you.
Thank you for being here. So please go with your presentation.
Yes, I will start sharing and see if it works.
So you have to share screen, the button on the bottom of your bar. Okay, it's coming. Then you put in presentation mode.
Yeah, is this okay?
Yeah, perfect. Thank you.
Great. Okay, thanks a lot for the invitation, and thanks a lot to the organizers for inviting me, for being on this conference, on this exciting conference. I think there were a few nice talks already. So today I will be, I'm Wim de Malse from the Free University of Brussels. And here you see, I think you see my cursor, so here behind the trees, here I have my lab. And so today I will be speaking about large-scale monodispersed droplet and particle production using 3D nozzle arrays. Okay. The talk will be mainly about droplet formation and subsequently particle formation. And so I'm sure you all know what an emulsion is. An emulsion is one phase of droplets, immiscible droplets within another phase. And a prominent example, especially in Belgium, this is an important example, is mayonnaise, where you have oil droplets within an aqueous continuous phase, which is stabilized by yolk, which is the surfactant. And so we can have a bunch of droplets that we create like this. And so what you see here is already very characteristic to the conventional way of making emulsions, is polydispersity. And so you see you have different sizes of droplets, and this is not always that great. For a bunch of applications, we would like to have monodispersed droplets, and there are applications, well, or possible applications, because this is not very much explored yet at the moment, but there are very interesting applications to be found in the assay world, in the analytical world, such as chromatography, for example. In cosmetics, there is a need for uniform particles for the same reason, more or less as in the pharmaceutical world, that you want to have a high control on the release of drugs or other active components from the vehicle in which it is carried. And so it's important that this vehicle and these release kinetics and what's inside is all very controllable. Other applications are in filtering, for example, in order to make uniform pores. In this place, in paint, the size is often related to the colors that are coming out of it, specialized food, and so on. So a wide range of applications, but very limited explored. So the end goal is typically making particles, and so a conventional way of making particles is by spray drying, where it has some limitations. A very common way of making droplets or making emulsions is simply by mechanical means, by having an impeller, which is mixing the one phase into the other phase and rupturing the droplets into smaller droplets maybe. But as said, this results in polydispersed systems. And so it's well known that already for 20 years or so that you can produce droplet by droplet under exactly the same condition, and this can lead to monodispersed solutions. So this is known. So my talk is not specifically about this aspect, but my talk is specifically about the main disadvantage of microfluidics, which is the very low throughput. And these conventional ways, they have a high throughput, they have a much more relevant throughput for many applications. And so we are working on bringing this low throughput to a relevant level. And I think these disadvantages, I've already told them. Maybe this one can be explained more in the context of you have more flexibility to manipulate particles and to manipulate the content you want to get inside particles if you can operate them one by one. So that's clear. You can have specific dedicated streams to bring drugs into, for example, into particles. And so you have some unique advantages if you can do one by one and if you can very controlled streams around particles. And so that's clearly also an advantage of microfluidics. And so one of the consequences of that is you have much more freedom also, for example, in tailoring the release kinetics. I'm not an expert in release kinetics, but I do, well, that's what I'm being told, that this is one of the problems at the moment. So making droplets, so when we look at an individual channel, first of all, making droplets can be done in a simple P-junction. And if this flow rate is relatively low, you make drop by drop, they touch the wall here, and you're in the so-called squeezing regime. And as you increase the flow rate, you get first to the dripping regime where you're not touching anymore. So this velocity is increasing, and so the droplets are pinched off earlier. And then you move into a so-called jetting regime, where this unstable blob of liquids will decompose. But the problem with the last case is here you will not have monodispersed droplets. So you will typically avoid going into this region. And doing so, people typically establish these curves wherein you can see the different regimes. And so then you will plot relevant dimensionless numbers, such as the capillary number, which is the ratio of the shear forces over the inertial forces. And then you can plot it against, for example, the velocity or the flow rate of the dispersed phase, and you can see that you can, as it increases, as the velocity of the continuous phase, for example, increases, you slowly run into these more unstable and more polydispersed regions. So these are things to take into account, because we will be pushing to higher flow rates. And so you cannot simply push to high flow rates because, well, without limits, because then you will run into unstable and polydispersed systems. Similar things can be done on flow focusing. So this is another way of making droplets. Also here you have similar curves you can make and similar phenomena that are occurring. And so how can we now increase the throughput? That's the main problem we are working on specifically for these kind of applications at the moment. So to develop reactors that are able to do this. And so one option is, okay, on the individual nozzle level, you can of course increase the frequency. That's not the topic of what I will discuss today. So there is some margin to work on that, but it will not lead to really industrial throughput if you're using a single nozzle. So that's limited. So the other aspect you need to work on is you need to combine or increase the number of junctions. And so you need to have multiple, and it's easily set, but not that easily done. And so there are different options to do this. One of them is in a so-called ladder architecture, where you have a main channel connecting to the individual nozzles where the droplets are formed. And then another option is that you have a central channel which is distributed, spreading to other channels. And so the main problem of both these approaches is that this is basically, these are two-dimensional approaches. So typically, this is a common microfluidic problem, you could say. This is a 2D type of system, and you will have channels in that 2D plane. So you have a depth, sometimes people call it 2.5D, but it's basically a 2D system. And so even if you only need, well, the location where the two droplets are being formed, the nozzle can be very small, but this whole channel here is actually the footprint that you need to make this single nozzle, and the same here. So all these connection channels are consuming a lot of place in your chip. And so in the end, you can parallelize a bit, but it's difficult to really get to very high throughput. And so the solution to this is what we've been working on now for a few years, is that instead of having these channels where you make where you form the droplets in the plane, we tilt it and we basically make these channels vertically. And so by doing so, you can have a plate which is perforated with many nozzles at a very high density. So then now we're not paying with big footprints to make each connection, because we make the connections in different layers. And so basically we're in the next layer and in the layer behind, we can make hierarchical structures with flow distributions. And then we have large channels connecting to many of the small channels. We can repeat this if needed, but we don't always do that. And so with this, we can, for example, have a 4 by 4 centimeter channel having 4,000 nozzles. And so this can, as you will see later on, this can produce really considerable flow rates. And so how do we evaluate our devices? We look from the side, basically, we work with syringe pumps or with pressurized vessels, air pressurized vessels, and then with high-speed camera, we look from the side and we look at the droplet formation. We are working on two types of substrates. One of them is plastics is, and these are typically PMMA, but also cycloolefin copolymer. These we are milling. So we are milling with our milling robot. This is very cheap. Only problem is we are a bit limited in the size of the nozzles, and so with the droplets. And so typically, for many of the chemical applications we are performing, this is a very relevant size. And also for many of the applications, the droplets shrink afterwards. And so they can still shrink tenfold, and then we can end up with 20 micron particles still. And that's a very relevant size for many applications. The other end is much smaller. I will come back to this in a little while, where we're using silicon glass chips, which are microfabricated, which use lithography, deep reactive ion etching, and advanced silicon fabrication methodologies. Right, and so this is a typical experiment where we're looking at the nozzle, inside the nozzle from the side. And so as you see here, this is a PMMA chip. You look inside, you have relatively large channels here, to order of 200 micrometers, and then these droplets are being formed. And so this is, to give you an idea of the speed, this is about 10 droplets per second that we are seeing here. And then you can plot here at this length is the unstable region that occurs in a drop that is in a droplet after a previous droplet was being formed. And so this gives you an indication of instability. So as this length scale increases, you get more and more to unstable regions, and you get into jetting regime in the end. And so as said before, when we are increasing the flow rate, we cannot just do this unlimitedly. There is one limitation is pressure, but the other one is we're going into unstable regimes. And pressure is in fact not really limiting for us. We typically only work below 5 bar. So for all the work we're doing, 5 bar is enough, but we could go to 100 bar if needed. Even for chromatography applications, we apply up to 400 bar on our chips. So in general, pressure limitations are not very relevant, but what is a limitation is you don't want to go into crazy unstable regimes. And so then you can also look at the lengths of these droplets then. So this gives you a measure of monodispersity. So you can see that as we go in the transition regime towards jetting, that we start to have bigger fluctuations in terms of size. And so here on the right hand, we also see the frequency, which is increasing as we increase the flow rates. All right. And so this is a typical image where we have this towards the unstable jetting regime. Now, if coming back to this pressure drop, so what you see here, if we have a single nozzle 5 ml per hour, then you can see that we are at 1 millibar more or less on pressure drop. But what is interesting here, and this was not the first design that we made, I don't have time to go too deep into it, but what you can see here is that so this is the first channel, the second, the third, the last for one of the phases. And you can see that we have the highest pressure drop in the last branch. And this is interesting, this is relevant, because it gives us a forgiving effect on fouling. And that is highly relevant, well, as we are making particles out of our droplets. We are having solid materials. Sometimes we have uncontrolled precipitation. So this is something to take into account. And so looking at this, studying this, what you can see here, these are again the dimensions of the different layers going smaller and smaller. And here the final channel, so the depth is 500 micron, the first width is 1 millimeter, final width is 500 micron. If we include defects now, so here this channel, these are 16 nozzles that we have here. If we include now one defect, we took away here a branch and we took away one here, you can see that the other branch will give higher velocities. These are average velocities across the exiting hole, you can see. And so these are aberrant flow rates here. And so these will produce larger sized droplets, as you could see a few slides ago. And so this is a problem. And so when fouling occurs, this is a problem. How can you cope with that? Well, by doing what I said on the previous slide, you can make sure that the highest pressure drop is in this very last channel. How do we do that here? Well, by, for example, decreasing the dimension of this last channel by a factor of 5. So you can see now all of these channels, including these two ones, they have exactly the same velocities. And so we really took away this fouling problem. And so this is one of one of the aspects. It's not a very sexy topic to work on or to publish on, but it's a very critical topic to get your devices to work. And here outside of the PMMA chips, you see droplets being formed here. Here again, we have 10 droplets per second, more or less. Here we have an array. So here we have 4 nozzles. We can only focus. We can only get 2 in the image, and so we have also 2 behind. You can see the droplets there, but we are making monodispersed droplets here. All of them, we can collect them. And then to give you an impression of the throughput, so here we're plotting the number of nozzles against the throughput. So if we look for, throughput and footprint, because we put, and you can see we have a lot of space left, so we in fact are totally not pushing yet the density of the nozzles, but having a density like this, we can get close to 10 liters per hour on a 1 square centimeter device. And so if you tell this to microfluidics people, this is a very relevant throughput. And you can easily increase this tenfold, and then you have a single wafer or a single substrate on which you would be doing this. So these are really very relevant throughput values for medical or pharmaceutical applications, for example. And as said, so the milling was kind of easy. You just mill your channels. If it's micro machining, we need to have multiple layers that we do lithography on. So we have several lithography masks. We do lithography. We have multiple layers. We are etching in the silicon, and so then we are etching first on the first device, and we're etching on the second device. And so to have the continuous phase flow distribution, and the discontinuous phase flow distribution. And then also we need to have a cover layer on the last one, and that you can see here. So we have glass on silicon on silicon. This is quite a complex and expensive fabrication scheme. And then we need to carefully make sure that we make side connections. So we need to make sure that we have a smooth cleavage over the substrate so that we can connect from the side. And so all this is quite some technology development, but we succeeded in doing this. So this is a characteristic image having many designs on it. This is a characteristic image. I think this is a 50 micron hole, if I'm not wrong. So here we are producing droplets about so much smaller than 200 micron. But basically, we can go up to a few micron. We produce droplets, I think, down to 20 micron or so. But so we are slowly evolving towards smaller and smaller droplets and hence particles. And I also need to add, I'm here now in the nanoparticle session, so we're not producing nanoparticles. So it's clear, we cannot produce nanoparticles using this kind of technology. So it's really out of scope. It's impossible. And this is again a similar type of image. We look from the side, we see multiple nozzles, and we are producing very more dispersed droplets over these different arrays. Now, another option that we often use, so this is using cheaper technology. Here we simply put a capillary inside another capillary. This capillary is sometimes a coned capillary, then we can make smaller droplets. That's the detail. But with these, this is very interesting to test different kinds of chemistries, because very often we have fouling problems, and we prefer to have this fouling problem then in a single capillary, replace the capillary and move on, and then afterwards testing our final protocol in our more expensive silicon devices. And also we have also made a parallelization of these capillaries into a 6 nozzle system, so that we can also produce relevant throughput in these capillaries. Now, this has been discussed already by the previous speaker, and also by the first speaker of the afternoon. And so, why are we interested in polylactic glycolic acid? Well, it's an FDA-approved material, and it's relevant for a bunch of applications. And I won't give too much attention to it, but we are specifically interested, and that's because of our collaboration with my colleague at the university. We are specifically interested in cell therapy for diabetes. And so what do? We make particles, so not nanoparticles, but microparticles, in which we put a drug. And that drug is being used to inject into the pancreas together with beta cells and to help those beta cells with the help of dexamethasone. So that's the help, and the previous speaker was also discussing it. We are using dexamethasone to help and to assist and to induce proliferation of the beta cells. And we are interested in producing particles of 10 to 50 micrometers. So that's what my collaborator, Professor Hellemans, at British Research Center has experience with our interesting particles, and she's using these now to do experiments, to do these experiments. These protocols have been known for a few years already how to produce these particles, but similarly as shown before, so these are polydispersed particles. The problem is these have not given very great results because of the unpredictable release kinetics, and they're not very stable. So that's an issue for my collaborator. So when we produce them, as you see here, they can be very monodispersed, and yeah, so this is an advantage. Well, these images now are PLGA particles already loaded with dexamethasone, and so this is well, these are they changed a bit because of the sample prep, the SEM. But you can see here typically, so this is a typical size range from 33 micron to 38 micron, which is produced in 6 nozzles, which is a monodispersed sample, and so which is the type of sample that my collaborator needs to make the conclusions that she wants to make. I also want to add that depending on the concentration of the PLGA that we use, if you use lower amounts of PLGA, during the formation of the particle, the liquid is extracted by an anti-solvent extraction, and so the less concentration of PLGA, the more the particle shrinks. And so you can see that we can, even starting from droplets of 80 or 100 micron, we can go down to 10 to 20 micron particles just by playing with the concentration. And so there's also quite some flexibility there, and so that's another parameter you can we can play with, we can optimize. But so this dexamethasone research is ongoing. I cannot really give you too many results on the medical applications, so there are no conclusions yet. But there is also another applications we are pursuing. Again, exploiting the fact that we can make monodispersed droplets and particles. This is together with the textile company. So this textile company is producing capsules, particles that are applied on a mattress, and they contain a bacterium, a type of bacterium, and this bacterium has anti-allergenic action. And so what happens is those capsules, when you lie on your mattress, those capsules, they will break. Yeah, but they need to break on a kind of controlled way, and they need to break over a period of a few months, for example. And so they cannot break. I mean, so you need to have very controllable rupture behavior. And for this, you need to have very controllable sizes and very controllable properties in general. And so what you see here, this is this contains a particle where the capsule is intact. This contains particles where the capsules were broken. These are particles with spores inside, and so you can see here there is no growth. Here there is growth, and so it's well known by the company that bacteria then has the anti-allergenic action, and so this is a highly promising result for the product that they are developing. So this was another little off-topic result that I wanted to share with you. All right, so that brings me to my conclusions. So we have validated a bunch of prototypes of the microfluidic methods able to produce droplets at high throughputs. Production of the droplets in the third dimension allows for the highest nozzle density. This is really critical to go to the tens or even the hundreds of liters per day. We have 2 fabrication methods. For the large particles, we have relatively cheap methods. For the smaller particles, we have more expensive devices, but again, we have high-value products, so this is in the end still quite okay. We can produce PLGA particles down to 30 micron, but we can reduce them by going to silicon, but also by playing with PLGA concentration. And then we have a wide range of applications we are currently developing. I would like to thank my co-authors of this paper and people of the group, especially Ilyas Bihi, who did most of the work here, who made most of the slides that I presented today. Philippe Legains, the valorization manager of our group. And I also want to thank the university for providing us specific funds for valorization, assisting us in doing specific valorization. This is not that easy to get this kind of funding at universities. And then Ilias is supported currently by Innoviris on his project in the context of diabetes treatments. And previously we were also supported by Catalysti and Vlaio and other related projects that were using the 3D emulsifier. And with this, I would like to thank you for your kind attention. Okay.
Thank you, Wim. Can you hear? Yeah?
Yes, I can hear you.
Okay, thank you very much. This was very interesting presentation, and for my side, having such uniform sites for particles is very useful in the biomedical field, where the release is related to the particle size, for example. So I have a question. So how low can you go with your sites using your method, and which is the density of these particles in your secondary flow in your liquid? So is it very consuming for the second phase? I mean, it's very big to have an amount of particles, or it's very dense, so we can also have a lower amount of solvent?
Well, it's a good point. And so, first of all, the continuous phase, we often use the continuous phase to get smaller particles, but then we have a lot of continuous phase. And so we are also working on, but you don't have to. So if you have relatively large particles for your dimension, because you can also play with your design of your microfluidic devices, of course. But okay, taking that, if you want to push it to very small particles, for example, or very small droplets, you will have a high continuous phase, and so you will have indeed a large dilution of your droplets and particles. But for this, there are a bunch of options. After formation, you can take out again your continuous phase. That's one option. You can do flow focusing. We are doing acoustic focusing. So there are many options to very rapidly concentrate again without spoiling your continuous phase. But I think, well, a very common concentration in volume percent, and maybe a typical volume percent would maybe 5 to 10% or so. I think that would be a typical value, but as said, we can make from that 5 to 10%, we can make 80% if we want. But that's not at this moment. That's not really a problem at this moment for us.
Okay, thank you. Maybe is there any question? Yes, we have one from Dario Puppi. Please.
Hi, thank you. Really nice presentation. I have just a curiosity. In case I would fabricate a microfilament instead of microparticles, can I exploit the jetting phase? I mean, if I want to obtain a microfilament of PLGA.
And with microfilament, you mean a kind of anisotropic particle you would like to produce, right? And with filament, you mean a very highly extended particle.
So it's continuous jet.
Yeah.
To have like a yarn or filament coming through.
Yeah, if I use your oil and water emulsion with the PLGA in an organic solvent, and I work in the jetting phase, do I obtain a continuous polymeric filament instead of microparticles? That's the question.
Yeah, no, I get your question. Thanks for the interesting question. So it's a matter of time scales. So I think the time scale of the instability is smaller than the time scale of the solidification of your PLGA particle. And so I think that wouldn't work, but I think it shouldn't be that complex. You could have, I mean, you have these segmented flow type of systems where you have a droplet followed by another droplet, and you could have channels next to that where you can still have your extraction. And I think you need to keep your droplet in an extended form. And so what you could do is, for example, you could have a droplet and you bring it to a narrow channel so that it remains an extended droplet. And then, and you could probably even have a sort of gradient that it gets narrower and narrower because your droplet will lose solvent and more and more solvent. So I'm sure there are options to do this, but probably not exactly on the way I show you today. So I think you would need walls to stabilize your droplet shape, basically. I think that's the bottom line. Because else, by capillary action, you will have it will want to go to a spherical shape, and you kind of need to, with having borders, you need to bring it into you need to keep it in an extended shape, basically.
Okay, thank you. Thank you.
Thank you.
Okay. So thank you very much. So I think we approach the end of the conference for today, if no other questions. So we thank Wim for his interesting presentation. I think there are several applications for this technology that can be exploited. So it was very interesting for us. So now I close the session in room A, and the other room is already closed. So thank you very much, and we see you tomorrow for the second day. And Wim, you can follow on YouTube channel. Okay. Thank you very much. Okay.
Thank you. Have a nice day.
Thank you. Thank you, everybody, for being here. And I want to remind, in your bag, you have the polybioskin survey. So please, because maybe you are not fully informed in the reception, please fill the questionnaire and return to the reception by tomorrow. So this would be very helpful for us to understand something about this project that is finished in this month. So for the social dinner, we have Trattoria Il Campano for the people who booked. And we can see tomorrow here. So same procedure, so maybe much easier tomorrow for everybody. And thank you very much for being here, for the help, for the understanding and everything. So thank you very much.